I research H-iPSC-derived Chromaffin-like cells transplantation as a strategy for relieving chronic neuropathic pain associated with spinal cord injury. SCI occurs when there is damage to any part of the spinal cord or the nerves at the end of the spinal canal. Following this damage, pain occurs, but if it persists for over 12 weeks, it is characterized as chronic pain. There are many different treatments to SCI, but the most common is standard pharmacotherapies or opioids. Though opioids do relieve pain, they are not ideal due to their short half-life that can lead to repeated use and potential misuse and addiction. There is a need for a new approach, and cell transplants have a high potential for being this new approach because of their ability to have a targeted delivery of pain-relieving substances, thus allowing for off-target side effects and standard systematic drugs to be avoided. The cell transplant that I chose are chromaffin cells, because chromaffin cells are secretory cells and produce a synergistic cocktail of analgesic or pain-relieving substances such as norepinephrine and metencaphalin. Because of how endogenous chromaffin cells are harvested, they have a low clinical feasibility in this form, but recent developments in cell technology have led to the development of human-induced pluripotent stem cells, or HiPSCs, which allow for chromaffin cells to be differentiated from pluripotent stem cells and a chromaffin treatment to be reevaluated. In addition to norepinephrine and metencaphalin, chromaffin cells also naturally produce a peptide called histogranin, which has a stable analog called serine histogranin, or SHG, but holds all the properties of histogranin, such as being able to reduce neuronal hyperexcitability that leads to reducing the physiological effect of pain. This leads me to my first research question. Are H-iPSC-derived Chromaffin-like cells pharmacologically similar to endogenous chromaffin cells and may serve as an analgesic for pain reduction? My hypothesis is stem cell-derived chromaffin cells releasing SHG may increase the analgesic potency of the cellular treatment. To evaluate this question and hypothesis, I performed a stimulation protocol. I first performed a baseline reading with Hank's buffer saline solution to have a baseline for the cell's norepinephrine and metencaphalin secretion. Then I stimulated one column of cells with nicotine solution and the other column with a nicotine plus mecamylamine solution. And then I performed a post-stimulation reading. All of these samples were evaluated with norepinephrine and metencaphalin ELISA assays. The results from both the norepinephrine and metencaphalin ELISA show a spike formation with the stimulation secretion levels being almost 3 times for norepinephrine and 2 times for metencaphalin compared to the baseline secretion levels, which mirrors endogenous chromaffin cells' response to the nicotine solution. However, the nicotine plus mecamylamine solution did not produce the spike. This is due to the fact that mecamylamine is a non-competitive inhibitor to nicotine at the nicotine acetylcholine receptor and prevents a spike in norepinephrine and metencaphalin production, as demonstrated by this figure. Then I confirmed that the H-iPSC chromaffin cells have similar pain mechanisms to endogenous chromaffin cells. I wanted to investigate if the treatment's analgesic potency could be increased, which led me to the second part of my project. My project investigates whether the SHG peptide localizes to the area of interest where it was introduced after injury, and do the SHG-producing cells have an increased analgesic effect in comparison to the H-iPSC chromaffin cells not genetically modified. My hypothesis is that H-iPSC chromaffin cells releasing SHG will localize to the area of interest and increase the analgesic potency of the treatment. The second part of my study, comparing genetically modified to not genetically modified chromaffin cells to stem cells, I completed immunofluorescence staining on genetically modified cells for chromogranin A, SHG, and DAPI location markers. From the immunofluorescence staining of the cell line, visualize the location of the chromogranin A, SHG, and the merged effect of the chromaffin cells and SHG. This exemplifies the potential to enhance the analgesic potency of the transplanted chromaffin cells using SHG. The method I performed was a fluorescent-linked immunosorbent assay, or FLISA. Prior to my joining of the lab, spinal cord and brain tissues from rats previously were intrathecally administered with either stem cell-derived chromaffin-like cells releasing SHG, the experimental treatment, or stem cell-derived chromaffin cells that did not release SHG, the control treatment. A set of tissues came from control treatment rats from a previous study, and another set of tissues came from experimental rats from the same previous study. I ran a FLISA to evaluate the optical density, which mirrors the concentration of SHG in the spinal cord and brain tissue samples. Their average concentration of SHG in the experimental spinal cord samples were observed in comparison to experimental brain samples. When an ANOVA was run to compare the experimental brain and spinal cord samples, a statistical significance was found. However, there was not a statistical significance between the experimental brain samples and the control samples. This confirms that the treatment was prevented from diffusing into the central nervous system, which makes the treatment more powerful in the region of interest and localizes the treatment. Solutions are the localization of the SCI treatment initiates continuous secretion of pain-relieving peptides in the spinal cord, and SCI-producing H-iPSC Chromaffin cells can be a viable alternative to opioids. In the future, I hope to compare this treatment's efficacy to that of opioids and test its clinical feasibility. Thank you.